Now, if we talk about a microscopic cell, we all know cells are microscopic in nature. A microscopic cell may also have thousands of, or you can say hundreds of reactions going on at one time, at a single time. So many reactions are going on even in a small microscopic cell. So that means there are enzymes required in these cells. And then the next point is that some of the reactions are like always going on. For example, we all know respiration is a continuous process. It has to be done every time. So like that means respiration can be considered as a reaction which is going on 24/7. So because it is going on every time inside a cell, then the enzymes required for such a reaction also are synthesized every time because they are used again and again. That means some enzymes are called as housekeeping enzymes, or you can say constitutive enzymes, because their services are required every time. Example, all respiratory enzymes can be considered as the housekeeping enzymes. So these are those enzymes which are essential and they are used in the cell every time. So always present in the cell, as they are involved in some essential or central metabolic activities of the cell. Example, glycolysis, which is common to both prokaryotes as well as the eukaryotes. So they are called as constitutive enzymes or housekeeping enzymes. Then another category possible is the regulated enzyme. Now those enzymes which are not required every time. So such reactions are called as regulated reactions, and such genes are called as regulated genes also. And they are because genes will form enzymes, so such enzymes are also not prepared always. They are only prepared when it is required. So that's why they are called as regulated enzymes. Now regulated enzymes are of two types. Now there are two things here to understand. There are some enzymes which are always required to be there to function, but if there is a condition, like for example product is in extra amount, it's something like, imagine for a second that we all have kitchen at home, and the ladies in the kitchen are going to function, they are going to be performing cooking and all every time. But if there is extra storage of food in the refrigerator, or if there is an invitation from the neighbors for a party or for some function and all, then obviously we are not going to cook in the kitchen. So it's the same thing. There are some enzymes, there are some reactions which are always going on, but if there is availability of the product of that reaction from outside, or if there is excess of the product present inside the cell, so the cell is going to go for switching off the reaction, and such reactions are called as repressible. That means they are always working, they are always switched on, but if there is a requirement, they can be switched off. Such type of reactions are called as repressible reactions, and such enzymes will also be considered as the repressible. So when there is a requirement, they can be switched off, but generally they are always switched on. And example, the example tryptophan synthetase in the E. coli, you'll be learning about these in the 12th class, because details of these topic come in genetics. And then in the same manner, you can have even the lactose digesting enzymes in the E. coli, or you can have nitrate reductase enzymes in the plants. These are examples of inducible type of enzyme. Now what is inducible? Let's understand. There are some reactions which only function when something is there to be reacted upon. So if there are substrate, if there are biomolecules on which reaction has to be performed, then only these reactions become active, or you can say then only these enzymes will become active. Otherwise these machinery will be always switched off. That means there are two categories. See, one is housekeeping gene, always working. Other are called regulated genes, or you can say housekeeping enzymes, always prepared and working. Regulated enzymes are those enzymes which are either always switch on, and when required they can be switched off. And then there are enzymes which are always switch off. They only function when raw material is there for them to function. So they are called as inducible. Inducible means they can be motivated, so they can be induced to function. That means they need to have an activator and they need to be repressed. Activator, what is activator here? The presence of the reactant. If reactant is there, enzyme will start functioning and the reaction will occur. I hope these 2 points are clear to you. So now, how enzymes function, let's understand this. Enzymes always act upon either the substrate or act upon the reactant. Now, be clear with the concept, when do you use reactant and when do you use substrate. When there is a single biomolecule, that means single type of biomolecule which is converted into another product, so you call such type of molecules as the substrate. But when there are two molecules reacting together in the presence of enzyme, then undergoing the change to form one product or maybe making two products, then you call those molecules as reactants. So single substance acted upon by the enzyme is called as substrate, and if there are more than one molecule involved in the reaction, then these are called as the reactants. Like you'll be learning many examples of these in the human physiology and plant physiology. So now remember, product word will be common whether it is this type of reaction or these reaction, and these reactions are controlled by enzymes.